Happy Thursday! Okay, it's time to do some biology. I hope you guys are ready. Here is my regularly scheduled tech check. If you can hear me, please say hi in the chat box so that I know I'm not talking to nobody, because that makes me feel awkward and self-conscious. Someone tell me hi if you can hear me. Pretty please. If you can't hear me, raise your hand. I can't see it. Maybe I'll telepathically get it. So if you can hear me, please say hi. Hi, Sandra. Nice to see you again. Yay. Ooh. Hi, Elena. Oh, my favorite people are here. Elena, Sandra, Davitha, and William. I'm so glad you guys are here. Woohoo. All right. Well, now that I know we're here, we're going to go ahead and talk about some membrane transport. So I hope you're ready. It's going to be super exciting. I know you're all very concerned with how stuff gets in and out of your cells because, you know, you got trillions of them. They got lots of stuff coming in and out and in and out and in and out. So we want to make sure we know how all of that works. Not only because it's a semi-significant portion of your AP exam, but because this is your body. How is stuff getting in and out of the cells of your body, and how is your body using it? How is that keeping you healthy? Lots of important things here, kids. So, as usual, please keep in mind that we love when people follow us on Twitter and Instagram and YouTube. You know, we're all millennials here, so we get our self-efficacy and personal value based on how many followers we have on the internet. So please follow those. If you do anything cool, like study, or you're like, hey, look at me, I'm being really cool, feel free to tag us because we may share you and therefore try to help you get some extra followers too. My name is Jessica Nadsam. I work at Jensborough High School. Normally I don't wear t-shirts on Thursdays, but grades are due this week, so t-shirts are necessary. So please follow me. If you don't already, I'd love to tell you some of the cool stuff we're doing. So today, we're gonna do this cute little introduction where I have this spiel and song and dance that I do for you guys every week or two. We're gonna review membrane structure because I wanna make sure that everyone here is on top of it, because if you don't know membrane structure, you're gonna be real confused about this membrane transport. So I wanna make sure we're all on the same page. Then we're gonna talk about selective permeability, so how things go through the membrane and how some things don't go through the membrane. They get stuck, like they hit a wall. We're going to talk about passive transport. We're going to talk about active transport. We're also going to throw in some bulk transport, just when we move big things in and out of the cell. So, hope you're ready. I apologize in advance. I know my screen is a little dark. The one lamp that I have, I knocked it over this weekend and it broke. So I have to get a new lamp because this is my lamp. But you didn't come here for my face. You came for my voice and my PowerPoint slides. Let's get real. That's all you're going to want at the end anyway. So here we go. All right, membrane structure. If you were here with me last week, shout out to Sandra and Elena, then you've already seen some of these GIFs, however you say it. I don't care how you say it, that's what they are. But you've already seen these before. So going back to very, very basics, what is this little thing in the top left? What's this thing that makes up the whole plasma membrane? What is that thing? Tell me. Tell me your secrets. Phospholipid, very good, Elena. So this is our phospholipid, and there's a very important quality of that phospholipid, and it is its polarity, like a polar bear. This unit is just an excuse for me to throw pictures of polar bears and seals and penguins and stuff everywhere, because it's polar. It lives in the North Pole. Because we have one end that has a certain charge and another end that has a different charge. Now, if you're a phospholipid, there is something called amphipathic. One end is having that hydrophobic or that polar charge. One end is actually nonpolar. Refresh my memory. Which end of the phospholipid is nonpolar? Tell me things. Which end of the phospholipid is nonpolar? Think, think, think. Think, think, think. The tails, very good. The tails are nonpolar, and as a result, if you look at this image, you can not really see it so much here, but you'll see it more in other images. The tails are all in the middle of the membrane here, which is very wiggly, and I'd like to add that's a real thing. The membrane doesn't just sit there pretty and posed. It's like not like the British guard who guard like Buckingham Palace, you know, they sit there with their hats and they don't move. And occasionally they march like every 12 hours. It's not like that. They're not still the whole time. Phospholipids are bouncing around all over the place because they have charges that they have to match. So phospholipids aren't going to be still. They have the tails, which are in the center because that protects that non-polar layer. And we want to keep it away from water because floating around out here and out here in this alien landscape, there are, in fact, polar things that the tails would not like. So we have the heads, that's this kind of yellowy portion on the outside, and that's what's actually these proteins and other molecules are bouncing off of. We're going to talk about that in a minute. But just to remind you that polarity is going to be super important to the structure, which is going to be super important to the function. So let's talk about how this is a selectively permeable membrane. When I say it's selective, I want you to think about that terrifying thing you keep putting off your college applications, unless you're a senior, which I really hope you've already done them. But if you think of like, say, a college application, most colleges, you think of them as being selective. Do colleges let everyone in who applies? No, unfortunately they do not. Depending on your grades, you may get in, you may not. It is a selective process. Selective permeability. Permeability means some stuff gets through, some stuff passes through. But since it's selective, not all of it gets through. So for example, Harvard is much more selectively permeable than say your local community college, which is much more permeable. I may have gotten that backwards. Harvard is going to be way less permeable. Not as many people are going to get into Harvard as say your local community college, where a lot more people may get in. Not saying there's anything better about Harvard or anything worse about a community college. They both have their perks. But there's a different rate of permeability in who can get in. That being said, one is also way more expensive, which that's a problem for a lot of people. So there are perks, there are not perks. Keep that in mind. This is not a college counseling session. This is simply a metaphor about selective permeability because not everything is going to be able to get through. And that's how it works with the membrane. So again, like I was saying earlier, the membranes, they're like Energizer bunnies. They're dancing, they're moving. Those phospholipids do not stay still. They are not statues. They're like me around 11 a.m. when I drink a pot of coffee and no one's in the room to entertain me and or for me to teach at. And so you just go and typing, because I have a lot of energy. Phospholipids have a lot of energy. They're constantly shifting and moving, and that's good because as they move, certain things, selective things, can pass through that membrane. We can see it happening in this GIF here. There's this one little molecule that can pass through. That's great because there are some things that we need to pass through. Oxygen, for example, it's pretty important. I need that to pass through the membrane. If I don't have oxygen, I get extremely cranky and then I pass out. So I imagine you guys are the same way. We've got to have oxygen. So certain molecules are going to have a much easier time of getting through a membrane. So I've got a little model here and it's got a synthetic lipid bilayer. So here's our lipids, just pretend it's gray bars instead of these very detailed wobbly phospholipids that I've got right here. But we have our synthetic bars. Some stuff is getting through, some stuff is not getting through. Do you notice anything in common about the kind of stuff that is not getting through? What do you notice about the stuff that has an arrow that just goes like, boop, boomerang? It can't get through the membrane. What do we notice about that? What do we notice about that membrane? Which stuff is getting sent whoop, instead of going through the membrane? The large or charged, very good, Elena. Large and in-charge molecules don't pass through a membrane. If they're too big, they simply can't fit through these phospholipids. It's like trying to fit something that's large through a teeny tiny hole. It doesn't fit. The size is incompatible. but also the charge. If it's got a polar charge, or even more so, if it's an ion, it may be fine with the polar heads, but as you can see here, a bunch of these are bouncing off the membrane because they can touch the polar heads, they can't touch the nonpolar tails. It doesn't work. Once they reach those nonpolar tails, they say, eh, and then they send them back. Just pretend all your cells at once are screaming, eh, because they don't want stuff to pass through. You think I'm being ridiculous? It's true. The nonpolar tails of all of your cell membranes are currently screaming because there are polar things trying to pass through. It's great because on the one hand, the polar things aren't getting through, which are things that aren't supposed to get through that way. That's fine. We're going to talk about other ways for them to get through. But it's a good barrier. We want it to have that barrier. So this stuff selectively goes through, selectively does not go through. I will tell you, when I teach my classes, I make sure that they know these 4 things. So if you want to snap, or take some notes real quick, or come back to these slides later, the small hydrophobic stuff like oxygen, carbon dioxide, benzene, atmospheric nitrogen, they tend to go through the membrane pretty easily, which is good because we really want oxygen to go in and we really, really want carbon dioxide to go out. If it doesn't, we would have a problem. Small uncharged things like water, glycerol, ethanol, they tend to go through the membrane pretty easily because they're still small. They're polar, but they're tiny enough they can usually kind of sneak through, or they have other methods that we're going to talk about in a minute. But the big stuff like proteins, big sugar molecules, DNA, they're not going to make it. And tiny things like a single hydrogen, they're really not going to make it because they have that charge from losing that electron or gaining an electron. That charge is just going to be incompatible with the nonpolar tails. It's like 2 ends of a magnet that have the same charge coming together. You know, whenever you try to put them together, no matter how hard you try, they repel each other. That's all that they're doing, and it's not going to work, unfortunately. So let's talk about how this is actually going to work in practice. And one of the key things, your teacher may have already harped on this with you, is called the concentration gradient. And you look at this and you go, I don't know what this is, teacher. I don't know what this is. And I'll be perfectly honest, when I was in high school, concentration gradient, and I had no idea what she was talking about. I was like, a gradient? Is that like how the color changes over time and it bleeds through? Because at the time I thought I was going to be an artist. Lo and behold, that didn't work out. But there was something about the vocabulary that didn't click for me. For that reason, I really want to make sure that I'm getting this vocabulary through to you because I'd like to save you from my struggles that I've already gone through. There's no reason for both of us to go through those struggles. So let's be very, very clear. A gradient is when something changes over time. So you've ever seen a picture where the color seems like it's bleeding? It's really heavy on one end and then it gets thinner and lighter and more transparent as you go on. That's a gradient. Cool. Concentration gradient. That's the amount of something in a certain amount of space. It's very similar to density. So whenever I teach density, especially in a liquid, I always think of Kool-Aid powder, or better yet, the lemonade powder, like the Minute Maid. You know the kind. You had the tub of it. If your mom was super awesome, she got you the pink kind. I'm in my mid-20s and we still keep the pink lemonade in my house. So, but there's also 2 people living in this house, me and my husband. I make it the right way with lots and lots and lots of sugar in a single glass of water. My husband makes it with a single scoop of sugar in a big glass of water. His is less concentrated, and I'm so glad he likes it so that there's more sugar for me. But when things are more concentrated, that means they have more of something in a smaller amount of space. So the idea of the concentration gradient is that you start out and you have more of something and it slowly bleeds out to the rest of it. If you've ever seen someone with food coloring that they drop in water, it starts out, it just stays there, but over time it all diffuses, and that has to do with movement of molecules and physics and things I'm not going to get into because you're not here for physics. All you need to know is things move from a high concentration to a low concentration. All I mean by that is that things flow over time until they're balanced. If you've ever had one of those friends, or maybe you are that friend, who likes a little bit of yoga, or singing Kumbaya, or happy, balanced things. I'm not trying to call you out or your friends. But I will say in cells, and this is what I tell my kids, because this is kind of one of the secrets, things in biology like to be balanced. Think about it. Your body likes to be balanced at a very specific temperature. If it's not at that temperature or very close to it, you don't feel so great. Most of you probably haven't experienced this yet either, but your body likes to be balanced at a very certain blood pressure. I'm assuming most of you are in high school and haven't dealt with this yet, but if you have, you know exactly what I'm talking about. When your blood pressure is not at that proper level, you don't feel good because you're unbalanced. If your blood sugar ever is not at the proper level, is unbalanced, you don't feel well. What I'm trying to tell you is your body is constantly trying to live at this certain level, and that level is balanced. You may call it homeostasis or equilibrium, whatever you're calling it, they all mean the same thing. We like balance. We want to stay balanced. And it's not just living things. Non-living like parts, abiotic factors of the world like to do that too. We have gravity. Everything likes to be flattened out over the earth at a very certain level. If you've ever twirled a water bottle, that water is going to stay balanced level with the earth. Concentration gradient is the same. The reason that you move from that high to low is that it's trying to balance over two sides. It's trying to make sure that you eventually have the same concentration all throughout whatever container. Whether that container is a glass of Minute Maid lemonade, or your body holding your blood and your fluids, whatever it is, you want it to be balanced. And I guarantee you, if you ever see a question on an AP test, whatever answer you pick, it has to do with things being balanced. You'll figure it out. We'll get more into that when we get into like the homeostasis type chapters and feedback. But let's talk a little bit more about this concentration gradient. So, for example, I have all these G's. What does that G stand for? What are these big red molecules? What are those big red molecules? They're big and red. Glucose. Very good, Elena and Sandra. Very good. Okay, so they're glucose molecules. They're big molecules. We also have potassium and sodium, which are very important electrolytes. That's why they tell you to eat a banana after you work out. You need to have certain salts in your body. But what we notice here is we've got a ton of glucose out here on the outside of the cell. You can see here there's a label that says outside of the cell. There's a lot of glucose there. A lot of sugar. But on the inside of the cell, there's not very much sugar. Where is there a higher concentration of glucose? Where is there a higher concentration of glucose? Outside, very good. And remember, concentration gradient, how do we move from what to what? How do we move molecules? High to low, very good, Sandra. Very good, Elena, on outside. So as a result, these outside glucose want to move in because we want to have the same number of glucose on each side. That's what concentration gradient is talking about. It's the movement of particles from high concentration up here to low concentration until eventually they're balanced. And they can do yoga and do crow pose and all those other things in yoga that people post on Instagram that I can't do, but I admire it a lot because, like I said, millennial Instagram. So let's talk about, once we get that gradient down, let's talk about the mechanisms that actually move this stuff. So I'm going to talk about cell transport mechanisms. There's going to be two in particular, passive transport and active transport. Here's the metaphor I tell my kids, because again, millennial, I watch a lot of movies involving mean girls and this wonderful actress from Pitch Perfect. So let me give you my metaphor here. There's two types of aggression. There's passive aggression, and you know what it is. You may not be able to define it, but you can think of examples when you've seen it. Maybe when someone is a little more like, I don't know, that's what you really want, and you can tell they're mad at you, but they're acting like they're not mad at you. Passive aggression. Then there's active aggression. As you can see in this wonderful clip from Mean Girls, I'm not condoning violence for any parents who are watching over your shoulders. But there is such a thing as active aggression, where there is someone who is angry and fighting and kicking and screaming and all those things. Which one takes more physical energy, passive aggression or active aggression? Which one takes more energy, passive aggression or active aggression? Active. Very good, Sandra. Active aggression, very good, Elena, takes so much energy. Like Regina George here is running around, slamming her lacrosse thing at people. She's running. She's probably out of breath and a little sweaty. It's a lot of energy. Passive aggression, you're just like, whatever. Whatever. It's not as much energy, not physically. I'm sure there's a lot of mental and emotional energy that goes into that negativity, but it's not as much physical energy. That's how I compare active and passive transport. Active transport requires a ton of energy. You've got to climb up a mountain to do it. You have to go against the concentration gradient, which we'll get into in a moment. Passive transport doesn't take any energy. You're going along the concentration gradient. And as a result, you just kind of coast down the hill, which is great. Who doesn't want to coast down the hill? So we're going to talk more about those, but I wanted to go ahead and give that metaphor because Mean Girls relates to people. They just remember things. And now you're going to remember active aggression costs more energy. So active transport costs more energy. So let's talk about passive transport. Passive transport is going to be the one that does not take energy. It relies solely on the concentration gradient because, again, things don't like to be unbalanced. If you've ever stood on one foot for a long time, you probably didn't like it because you were unbalanced. If you've ever been sick and had a fever, you probably didn't like it because you were unbalanced. We want that homeostasis, and so do your cells. Your cells are what make you up. They're the things who create that balance the most. And so if we look at this cell, we're going to see it in 3 different phases. One where all of these blue hexagon molecules, let's just say they're sugar molecules because they're shaped like glucose. So let's say these are glucose molecules. Where is there a higher concentration of glucose in that first frame on the left? Where is there a higher concentration of glucose? At the top. Is that on the inside or the outside of the cell? Inside or outside? Very good, because it's extra, as in exit cellular, outside. Very good. And so over time, what we see is some of them are starting to pass through this membrane. And as time goes on, eventually they're all on the same playing field. There's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 on the extracellular side. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 on the intracellular side. It's in equilibrium, it's in homeostasis, it's balanced, it's happy. Now, here's the trick you have to know for AP. That doesn't mean that stuff is not crossing the membrane anymore. There is still traffic going both ways. But we say that there is no net movement. What that means is we're not suddenly getting 15 on this side and 13 on this side. They're still moving, but they're moving at a rate that keeps their balance and keeps them equal, and that's what they like. So passive transport, that's when molecules are just going to pass through the membrane like, here I am. It's like a hop, skip, and a jump across. It's easy. It doesn't cost any energy because they're just riding the concentration gradient. Facilitated diffusion works a lot like that too. Facilitated diffusion is a lot like, oh, am I out of order? Where'd I go? There we are. Okay, sorry, I double clicked. Facilitated diffusion is going to be a type of passive transport. So if you're taking notes right now, facilitated diffusion is a type of passive transport. Same thing is happening as before. I've got some molecules. In this case, I've got 2. I've got little green hexagons and some light green ovals. Yeah, those are ovals. And they're having to get in and out of the cell. Extracellular space, intracellular space. They are riding the concentration gradient to do that. But let's say, remind me again, what type of molecules? So it's passive because of that middle stage. Ooh, so Sandra, okay, let me fix this real quick. It's passive because it does not require any energy. So go back real quick. This is passive because it does not require energy. You do not have to pay ATP for it. It's not because of the middle stage. It's because it starts at high concentration and the molecules are moving towards the lower concentration. It moves from high to low. It moves along the concentration gradient. That's why it's passive. And we're going to talk about the comparison active in a minute. But did that answer your question, Sandra? No. Yes. Okay. Thank you, Sandra. Very good question, though. So it's passive because it does not take energy. And facilitated diffusion, like I was saying, it's another type of passive transport. No energy required. It's like a free ferry. And the ferry is this cute little protein. Because remember, in your membrane, you have what we call a fluid mosaic. You've got these phospholipids that are constantly moving around, and you have the proteins that stub it. So in facilitated diffusion, these are the molecules that are too large or too polar to actually pass through the membrane. Remember, we said some molecules, they're just too big. They literally don't fit, or they have a certain charge that's polar, and that polar charge would never be able to get past these hydrophobic tails. So they use proteins, which are able to fit between the membrane and just kind of drop things where they're supposed to go. Whether that's into the cell or out of the cell, that just depends on the concentration. Again, like we see here, there's a high concentration of molecules up here. There's way more molecules up here. There are less molecules down here. So it's moving from high through the proteins down to low. But since it uses proteins, it's facilitated. If it didn't have proteins, it would just be simple passive transport. Do I have any questions about facilitated diffusion? 3, 2, 1. Okay, I'm going to go ahead and move on to active transport. So this is the fun one because it costs money. This is when you got to put the gas in the car. You got to pay for that bus card. You've got to go against the concentration gradient. So we're going to say this dotted line is the cell membrane. Pretend it's the cell membrane. But now I've got these molecules, and do you see how there's less concentration on the right and more on the left? But you want to move molecules to the left. So you're moving from a lower concentration to a higher concentration. This is that active aggression we were talking about. It takes energy to do that because you are literally riding a bike up a 90-degree hill. It takes so much more energy to do that. And so they pay for it in a couple of different ways. The big one that you're probably going to talk about in your biology class, if you haven't already, is called the Sodium-Potassium Pump. This is like the classic example. It's like how you always hear about people dissecting frogs. That's the classic biology experiment. Sodium-Potassium Pump is the classic cell transport experiment, or example, not an experiment. We don't need an experiment. We just know it's there at this point. But so again, we've got a cell membrane, and we're looking at sodium and potassium. Sodium and potassium are electrolytes. If you've ever gotten a Charlie horse or a leg cramp, that means you didn't have enough of those. And as someone who regularly wakes up with them in the middle of the night because she drinks too much water but not enough electrolytes, it's not fun. You gotta have your electrolytes. Sodium, potassium. Eat a banana, sprinkle a little salt on your food. But we have to get these in and out of the cell because you don't want them both on the same side. That's why it's tricky. So let's take a look at this chart. So what you've got here is a very specific protein. And notice how it's got very specific shapes. On one side, it's got this pointier, like box shape. On this other side, it's got a more curvy shape. Remember, structure and function. Different structures, different shapes, different jobs. So in this example, you can see we've got a higher concentration of sodium on this side. These orange hexagons. And notice it's an Na+. What does that mean if it's got that plus on it? What does it mean if it's got that plus? It's charged. Very good. And Sandra, I just saw your comment, so it's moving from the inside to the outside. Not necessarily. Passive transport can move from the inside to the outside. It's not about where it's moving, it's about on the concentration gradient. Active transport, it moves from low concentration to high concentration. And that costs energy. So active transport costs energy, and it moves from low to high concentration. So we'll talk a little more about this through this example, but stop me if you have any more questions. I'd love to hear them. But so what we've got this sodium, and we've got this potassium, this round yellow egg thingy. That's what I'm going to call it. There's a low concentration of potassium out here, but there's a high concentration of sodium. But down here on the inside of the cell, I've got a high concentration of potassium, K, and a low concentration of sodium. Are those balanced? Are they equal? Are the sodium and potassium concentrations in equilibrium? No, they're not. And so they're not happy. They don't want to be like that. So we need to make them happy. So what we're going to do is we're going to move sodium out and potassium in. But that's going against the concentration gradients at some point, and that costs energy. So we can see here, we're actually moving sodium out and potassium is going in. That's not making it more balanced. That is against the concentration gradient. That's expensive, and that's why we have this thing. It's called ATP. If you haven't already talked about that in the cell, ATP is like cell money. If you want to get on a bus, you got to swipe your bus pass or give them a quarter or whatever. I live in a town that doesn't have buses. We don't have enough people for that. But if you want to take a mode of transportation, somehow you got to pay for it. Whether you're putting gas in your own car or paying for the bus or paying for the Uber or paying for the Lime scooter, whatever it is, you have to pay for it. Same thing for active transport, and they pay in ATP. So with ATP, these molecules are able to switch. So make your hands go like this. Follow me. I'm going to make my screen big for a minute here. All right, I want you to look at me. This is very important. And if you move your hands, you're going to remember it because that's a modality. I learned that in teacher school. It means it's real. Work with me. At some points, the protein looks like this. So it's closed up here, but open down here. I'm just going to assume you're all following along. I can't see you. But then when you get an ATP, so let's say my chin is the ATP, it goes ding, and the elevator door opens. And now it's open on the top and closed on the bottom. And that releases what was ever inside the protein. Now whatever's on the outside, which we know to be the sodium is all on the outside, can fill your protein. And the ATP hits it and it goes ding, and it opens up on the other side. So this protein is constantly shifting. It's like a one-way door that only lets in and out in some places. And it only lets two things in. It only lets sodium and potassium work one way. Exactly, Elena. It's a revolving door. Thank you. I was thinking that word and couldn't remember it because I've been up since 5 a.m. It's a revolving door. But it's even more than that because only some stuff goes in and some stuff goes out. So that's going to be critical through this too. So I'm going to pull that tab back up. So like I said, you've got some stuff that's coming out. It's trying to get all the potassium out and all the sodium in. And when your body doesn't have that, that's when you get a Charlie horse and your leg hurts and you have to get up and massage it and whatever. If you've ever had a Charlie horse, you know exactly what I'm talking about, and it is not fun. You have to get up, stand on it, massage it, and probably eat a banana. But that's active transport. It's moving against that gradient because we are not making this balanced. We are not at all. And that's costing us energy. It's costing that ATP. Do I have any questions about active transport? We're about to move on to the practice questions. Ooh. 3, 2, 1. All right, I'm going to move on. Oh, we do have one more thing. So this is kind of cool. This is a cell, and he's eating another cell. He's like that cell's worst nightmare. He is literally going chomp, chomp, big shark, I eat you. I'm sorry, I'm a nerd. This is really exciting for me. And when I found this on the internet, I was like, I have to show them. But you can see this cell just opens his big cell mouth and goes, and he eats the other cell. That would be an example of something we call endo, as in enter, endocytosis. And so literally what the cell does, it finds this foreign body, reaches out its big cell plasma membrane mouth, chews it up, so now it's on the inside, and then it uses some lysosomes and other enzymes and breaks it down. Now, on the other hand, we have something called exo. Exo meaning exit. Let's say it ate this thing and it didn't want it, and it wants to spit it back out. So, like, we got these little dots down here, exocytosis. I don't know, maybe it's a giant molecule it's got to send out. I don't know what it is. I can't think of any examples, I'm sorry. But it's saying, oops, exile. And so it puts a little vacuole around it and spits it out. That's exocytosis. So these, I think the endocytosis is going to be more common when you see a cell literally eating another cell. If you go on Jiffy or GitHub and you search cell biology, it's all these videos of just cells eating other cells, which it's been really hard not to put them all on the internet here so you guys can see. But endocytosis is going to be really common in cells when they consume other cells. You're actually going to hear about this later when we talk about endosymbiosis, which has to do with mitochondria, and it's fascinating. But exocytosis is going to be important too, because sometimes there are things you've just got to get out of that cell, because otherwise it could destroy the cell, and that would be bad. Any questions about bulk transport? I don't really have much more, just knowing what they are. There's not much more to them. Any more questions about bulk transport? I'm going to give you 3, 2, 1. All right, cool kids. I'm going to go ahead and move on then. Dun, 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 dun. Ooh, let's do a practice AP question. Okay, so one piece of vocab I didn't go over with you, simple diffusion. Simple diffusion is the same thing as passive transport. It's just moving down a concentration gradient from high concentration to low concentration. How are simple diffusion and facilitated diffusion related? Tell me your answers. Yes, the link will be provided to the Google Slides, yes. I see B, I see B. Well, I see 6 people attending. At least one more person give me an answer. Pretty please. It would make my day. It really would. 3, 2, 1. Okay, I wasn't trying to not draw attention to y'all. Sandra and Elena, very good job. Da-da-da. It's going to be B. It depends on that concentration gradient. For simple diffusion and facilitated diffusion, how do we want that concentration gradient to go? How do we want that concentration gradient to go? High to low. Very good. So Sandra, watch out for this out to in. It's not outside or inside. It's high concentration to low concentration. Because it could be high on outside or inside. It could be low on outside or inside. It's all about where it's highly concentrated. So like we've got a lot to where we've got a little. Like what if I flipped this and what if I got rid of this extracellular and flipped it with cytoplasm? Then I'd have a bunch on the inside. And that happens sometimes too. It's not about outside or inside. It's about high concentration to low concentration. Okay. Practice AP question number 2. The manner in which several different ions and molecules move through a cell membrane is shown in the diagram. It's not above, it's below, sorry. But we got this picture here. For each ion or molecule, the relative concentration on each side of the membrane is indicated. Which of the following accurately describes one of the movements taking place? So I've got high oxygen to low oxygen, high sodium to low sodium, high glucose to low glucose, high sodium and low sodium to low sodium and high sodium and low sodium to low potassium and high potassium. What do you think? Tell me your thoughts. You know things, tell them to me. D, I see D. And sodium transport out of the cell requires ATP hydrolysis. Why does sodium transport out of the cell? So from here in the cell to out of the cell, why is that requiring ATP? Why does that require ATP? Low to high, it's active. Elena, Sandra, great job. It's active. We are not moving along the concentration gradient. We are moving against it. We are rebelling. We are going from low to high. We are climbing the mountain instead of sliding down it. Very, very good. Any questions about practice question number 2? Three, two, one. Bueno. All right, last practice question. Which of the following scientific questions is most relevant to the model represented in the figure below? I've got some molecules moving all over the place. What do we think? Think, think, think. Brain, brain, brain. I see C. Give me one more answer, guys. Tell me your thoughts. Tell me what you're thinking. One more person. And C? Da-da-da-da! C! Which one is being transported across the membrane? We don't know that there's sugar. It doesn't say anywhere here that this is sugar, so it's not A. What types of phospholipids? There's only one type of phospholipid, and that's the type we use. It doesn't say anything about temperature, so very good. The answer is C. Awesome job, guys. So I'm sorry we're a little bit short tonight, but that's all I've got. There's not a ton of questions in my test bank in this, which I'm hoping I can expand that this year to get y'all more practice, but a lot of it's pretty straightforward. The main thing I will tell you to get tattooed inside your head, in your brain, on your skull is concentration gradient moves from high to low. That is passive transport. If it's moving against the concentration gradient, that's going to be active transport. It's moving from low to high. I'm going to go ahead and share these slides with you because I know that's why you're all here. But then I think we're going to be done, y'all. We're done a little early today. Oops. So la 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 la. Here we go. I'm gonna copy the link, send you the thing, because I'm so nice. Hope y'all like my singing voice. I see a question from Will, but it's not letting me see it. Hang on. My computer's being silly. I enjoyed your, thank you, Will. I appreciate that. Well, I will be back next week on Wednesday. My schedule is a little off. So next Wednesday, I will be back. I don't remember what I'm talking about, but I will be here and it's something in this unit. So hope to see you guys then. But until next time, y'all, go do some science. Have a great day. Thank you, Sandra. Hope you had a good time. Hope this helped. Bye, Elena. Bye, Davida. Bye, all people who were here. See y'all.